Hello everyone, welcome back to my YouTube channel. This is Soumya. In this video, I'm going to solve the KSET license question paper from 2014, and this is the part 6 video, and I'm solving the paper which is paper 3. Begin with the 58th one. Total genes of a given population is option A gene frequency, option B genotype, option C gene family, option D gene pool. There will be a population. Okay, think this is a given population. In this given population, how many genes are there? Okay, so like this many genes are there. Okay, so all these total means all these genes in this given population is called as what? Actually, this is called as option D is the correct answer. That is called as gene pool. Okay, the total genes of a given population, it is called as gene pool. Okay, moving on to the next question. 59th one. Use and disuse theory was proposed by option A Morgan, option B Lamarck, option C Darwin, option D Weismann. And before solving this, let us know what is this use and disuse theory. This use and disuse theory was proposed by Lamarck. So as in the question, they are asked who proposed this. So option B is the correct answer. This was proposed by Lamarck. And as in the name indicates, use and disuse. Use and disuse theory states that new needs arises in animals as a result of change in the environment, and this one actually leads to a new type of behavior involving new uses of pre-existing organs. Okay, and their use leads to an increase in the functioning, or you can say for example increase in the size, or okay, like that. And what disuse says, disuse means if you are not going to use that part any of your body organ, for example, okay, or if you are not using that for a longer time, that functioning of that part is going to decline. The functioning of that part is going to decrease. Okay, which one you are going to use more, that is going to work well. Okay, the method of functioning is going to increase, but when the part that is you are not going to use, that work efficiency or that part is going to decline. Okay, that actually means the use and disuse theory. It is a beautiful theory that is proposed by Lamarck, and actually he is the first person to propose a mechanism by which gradual change of species might take place. So how the species are changing? Species are changing gradually. Okay, that's what he proposed, and he also describes a two-part mechanism. What is this two-part mechanism? Two-part mechanism by which change was gradually introduced into the species, and those are passed down through the generation. Means those are that gradually changed characters in the species. Those will be passed on to the next generation. This theory is called as theory of transformation, or you can say just Lamarckism. Correct answer for this is option B. Okay, moving on to the next question, 60th one. Question is like this. Consider the following statements. The main drawbacks of Darwinism were option A, the lack of any direct evidence of the effectiveness of natural selection in nature. Second option, considering evolution as individual phenomenon, not as a population phenomenon. Third option, ignorance of law of inheritance. Option fourth, struggle for existence. So options nodona, which of the following combination is correct? Option A nalli option 1, 2 and 4th kottidare. Option B nalli first, third and fourth, and option C nalli first, second, third. Option D nalli second, third, fourth. Now first we will know Darwinism first. Okay, then we will move on to the drawbacks. What is this Darwinism says? Darwinism, it is also called as theory of biological evolution. Okay, so in this he talked about a natural selection, and then inherited variations, and he also talks about the phenotypic variation, and he also says about the struggle for existence. These actually the Darwinism's theories. Okay, and this struggle for existence is a concept in Darwinism theory. Okay, that actually refers to the competition between the species to survive and to avoid the extinction. Okay, that is called as struggle for existence. You should always fight to live, to survive. Okay, and who is the stronger one? Those are going to survive. Okay, that actually represents the struggle for existence. Okay, so this is the one point in Darwinism. Another point is he talks about the phenotypic variations. Okay, and he also talks about the natural selection. But here they are asking about the drawbacks. The main drawbacks of the Darwinism here is that the first option is also correct, because there is a lack of any direct evidence of the effectiveness of the natural selection. Yes, he says about the natural selection, but there are no direct evidence for that. So hagagi first one is the drawback. And coming to the second one, considering evolution as individual phenomenon, not as population phenomenon. Why Darwinism giving is Darwinism theory, he considered evolution as an individual phenomenon. See, he didn't take the whole population. This one is also another drawback. First one and second one both are drawbacks. And coming to the third one, ignorance of law of inheritance. Yes, he is not given any law of inheritance. How he says about the inheritance, he says about the inherited variations, but he doesn't say how it is going to inherit, and that's why he ignored the law of inheritance. So this one is also become the drawback of Darwinism. And coming to the fourth one, struggle for existence. Actually, it is a Darwinism one of the main point. Okay, like struggle for survival of the struggle for existence, and then phenotypic variation, and then natural selection, gradual changes, and inherited variations. These are all the theories in the Darwinism. Okay, so ಹಾಗಾಗಿ this is not the one. So ಇವು ಮೂರು ಯಾವ ಆಪ್ಷನ್ ಅಲ್ಲಿ ಇದೆ ಅಂತ ನೋಡೋಣ. That option there in the option C. So first, second and 3. Okay, third one. So these are the drawbacks of the Darwinism. Moving on to the 61th. All of the following are believed to contribute to genomic diversity among various species, except option A, gene duplication, option B, gene transcription, option C, lateral gene transfer, option D, chromosomal rearrangements. Before our answering this, we will know what is genomic diversity. Genomic diversity refers to it is a DNA sequence differences among the individuals, or you can say in the population. Okay, that is called as genomic diversity. Diversity means you can say different varieties. Okay, varieties of genetic characteristics. Okay, that actually represents the diversity. So genomic here refers to the DNA sequence difference among the individuals, or you can say among the individuals, or you can say among the population in a broader sense. Here they are asking which is not contributing to this genomic diversity anta ಹೇಳ್ತಾ ಇದ್ದಾರೆ. So option A, gene duplication. As in the name indicates, genes are going to duplicate. They are going to get doubled. Okay. So it means that there is an increase in the varieties, increase in the genomic diversity. That's why even option A is the correct one. Okay, option B I will explain this later. We will move on to the option C here. Lateral gene transfer. What is this lateral gene transfer? Lateral gene transfer is nothing but it is also called as horizontal gene transfer. This lateral gene transfer defined as is the movement of genetic material between organisms. If you come across any lateral gene transfer or a horizontal gene transfer, it is actually represents the transfer of genes, or you can say the movement of genetic material between the organisms. Movement of the genetic material between the organism, that is called as lateral gene transfer or horizontal gene transfer. Two types of gene transfer, that is lateral gene transfer, or you can say it has another name, that is called as horizontal gene transfer. And second type is vertical gene transfer. What is this vertical gene transfer? Vertical gene transfer is the inheritance of DNA from the parent organisms, and this is going to occur through sexual reproduction and asexual reproduction. And coming to the lateral gene transfer, here it is going to take place through the transformation, transduction, or you can say through the bacterial conjugation. This horizontal gene transfer, if I have to mention a key point or key difference between the lateral and vertical gene transfer, means lateral gene transfer, it is going to takes place between the unrelated organisms, and vertical, it is going to take place inherited from the parent organisms. Anyhow, according to this question, so here the genes are getting doubled and diversity is going to increase. So option C is also correct. And chromosomal rearrangement, obviously if the chromosome get rearranged, then you are going to get a varieties of genetic characteristics. So it includes for the genetic diversity or genomic diversity. Option C is also correct answer. So now the left one is option B. So option B is not the one. That is gene transcription. What is gene transcription? Transcription andre nimge already gottide enaagtade, just you are going to transcribing that gene. That's all. Okay, there is no you are not going to add any different varieties there. As it is there, you are going to copying that. That's it. Okay. So option B is not the one which is not including. So option B is the correct answer. Now moving on to the next one, 62nd question. Choose the correct combination of molecular markers used in PCR based DNA amplification. Option A, RF, LP, AF, LP and SSR. Option B, AF, LP, SSR and RAPD. Option C, RF, LP, RAPD and SSR. Option D, RAPD, RF, LP and SSR. Here they are asking for the correct combination of molecular markers which is used in the PCR based DNA amplification. Okay, the correct answer for this is option B is the correct answer. These are the molecular markers that are used in the PCR based DNA amplification. So what is this AFLP represents? AFLP represents amplified fragment length polymorphism. So amplified means here amplification means you are going to amplify, you are going to doubling the length here. Okay, that actually the meaning here. And then SSR represents the simple sequence repeats. Okay, and RAPD represents the random amplified polymorphic DNA. These are the molecules used in the PCR based DNA amplification. And what is this RFLP? RFLP is restriction fragment length polymorphism. These three are the ones which are PCR based techniques. Next, moving on to the next question. Choose the correct sequence of events in a next generation sequencing based whole genome sequencing project. Option A, DNA extraction, sharing, library preparation, sequencing, assembly, finishing, annotation, submission to gene bank. Option B, DNA extraction, library preparation, sequencing, assembly, annotation, finishing, submission to gene bank. Option C, DNA extraction, sharing, adaptation, ligation, library amplification, sequencing, assembly, finishing, annotation, submission to gene bank. Option D, DNA extraction, adaptation, adapter ligation, library amplification, sharing, sequencing, finishing, assembly, annotation, submission to gene bank. Before answering this, we will know what is this whole genome sequencing. Okay, whole genome sequencing, that is WGS. It refers to the construction of the complete nucleotide sequence of a genome. Means it refers to the construction of the complete nucleotide sequences of a genome. Complete nucleotide sequence now they are going to construct. That's why it is called as whole genome sequencing project. And how they are going to do that? So our steps na ಕೇಳ್ತಾ ಇದ್ರೆ one by one order ಅಲ್ಲಿ ಯಾವುದು ಕರೆಕ್ಟ್ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, in the first step in the whole genome sequencing project, what they will do, they are going to collect the sample first. Okay, and they are going to do the isolation. From there they are going to extracting the DNA. Okay, that is called as means they are going to prepare the sample. First they will do DNA extraction. So in all these four options, DNA extraction is the first one. After that they are going to do the fragmentation. Fragmentation means like they are going to do the cuttings. Like sharing means like they are going to cut the sample. That is there in option A and option. So you can leave out these two, option B and option D here. After cutting, next we will move on to the next step. And then later they are going to do the library preparation of the DNA. Means so after cutting they are going to prepare the library. They are going to prepare the copies of it. That is their library preparation. Okay, option A nallu ide, option D nallu ide. Next we will move on to the next step. Okay, then later they are going to prepare the sequencing. Next step is also sequencing. In this also it is correct, in this also it is correct.
So coming to option A ನಲ್ಲಿ ಏನಿದೆ, DNA extraction ಇದೆ, sharing ಇದೆ and library preparation ಇದೆ. But before after sharing there should be adapter ligation. So that step is missing out here. Hence option A is not the correct one.
So correct answer for this is option C, where DNA extraction is going to take place and they are going to cut the DNA and then they are going to do the adapter ligation. Okay, they are going to add the adapter to ligation means tying. Okay, so they are going to add the adapter ligation here and then they are going to do the library amplification, means they are going to amplify the content, and later they are going to do the sequencing. Sequencing, it is for the analysis purpose, and they are going to do the assembly and they are going to do the finishing, and then moving on to the annotation. Annotation is represents the process of describing the structure and function of the components of the genome by analyzing and by interpreting the sample. Okay, that is called as annotation, and then later they are doing the submission to the gene bank. Correct answer banbitu option C is the correct answer. Moving on to the next question, 64th question. Which of the following forms of DNA travel faster when run together in a cross gel electrophoresis? Option A, nicked supercoiled DNA. Option B, supercoiled DNA. Option C, linear DNA. Option D, plasmid DNA. And the correct answer for this is option B, that is supercoiled DNA. Because if you take this as a agro-based gel electrophoresis, so if it is supercoiled means when it is super compacted. Okay, so that one is going to flow fastly through this gel. Okay, they are going to move fastly, or they are going to travel faster when it is highly complicated, highly compacted. Okay, super coiled means compact sized. Hagagi that's why this super coiled DNA is going to move faster, not the linear DNA. If it is linear like this, it is difficult to move. Okay, and the nicked super coiled. Nicked means there is a gap, or there is a damage in the super coiled DNA. So if that is the case, it is unable to move faster. Correct answer is option B. Moving on to the next question. After agarose gel electroporosis, RNA is detected by option A horse radish peroxidase, option D glucose oxidase, option C cDNA probes, option D spectrophotometer. The correct answer for this is option C, that is they are going to detect the RNA using the cDNA probes. Next, the scintillation counters detect radioactivity by option A absorption of radiation, option B excitation of flows, option C ionization of gas by radiation, option D molecular size. First, we will know what is this scintillation counter. Scintillation counter, it is an instrument that is used for measuring the ionizing radiation. They are going to measure the ionizing radiation, means radioactivity, and it comprises, or it contains the scintillator that generate the photons in response to incident radiation. It is going to work on the excitation of the photons, based on the excitation of the photons, or you can say excitation of the flows. Option B is the correct answer for this. Moving on to the next question. Which of the following organism genome was chemically synthesized? Option A Mycoplasma genitalium, option B Saccharomyces cerevisiae, option C Rana pipianus, option D Drosophila melanogaster. The correct answer for this is option A, that is Mycoplasma genitalium. As in the name indicates, genitalium, that means which is something related to, this is a bacteria which actually affects the sexual part, that is genitalium. Okay, and it causes the sexually transmitted diseases as well, like it causes the itching and burning sensation. Okay, if that area is affected with this bacteria. Okay, and this bacteria is the one which can be chemically synthesized. Okay, option A is the correct answer. Moving on to the next question. The total protein coding sequence is the human genome is. Okay, total protein coding sequence in the human genome ಕೇಳ್ತಾ ಇರೋದು. The correct answer for this is option B, that is 1.5%. 69th question, they are asking here the match the following here. So you will look at the first list 1 and list 2. In list 1 we have desert adaptation, and in list 2 first one is gibbon, and in list 1 fossorial adaptation, and in list 2 naked mole rat is mentioned, and in list 1 third one is scansorial adaptation, and in list 2 it is opossum, and in list first fourth one is arboreal adaptation, and in list 2 fourth one is horned toad. Okay, desert adaptation. So which animal is adapted for the desert here? Here the desert adaptation, horned toad. Okay, so for this make you to understand, I will just display a pictures here. You can see this is your horned toad, actually adapted for the desert. First option here, fourth one correct agide. Next one, fossorial adaptation. What is this fossorial adaptation? Fossoria, the fossora means, this actually the word meaning is digger. I mean animals which live underground, or by digging the underground. Okay, those are called as fossorial adaptation. Okay, that is called as fossorial adaptation. Okay, which are going to live, animals which are live in the underground or by digging the soil. The option that is naked mole rat. Okay, this is the one which is going to live underground. So you can see in this diagram, this is called as naked mole rat. So this is the one which is living in the underground. And the next one, scansorial adaptation. What is this scansorial adaptation? Actually, the meaning of the scansorial means it is capable of climbing. The organisms, what they will do, the animals here, they can able to climb on the rocks, on the wall, and on the branches of the trees. And at the same time, they are going to living on the ground as well. Means here scansorial means they are going to live both on trees and also on the ground as well. So they are going to live on the tree and as well on the ground. That type of animals are called as scansorial animals, or you can say scansorial adaptation. And here that is done by a opossum. Okay, I will show the diagram of this. You can see this opossum. This animal is going to live on the ground and as well as on the tree as well. Okay, that's why it shows this scansorial adaptation. And the last one, arboreal. What is this arboreal adaptation? Arboreal adaptation represents the animals which spend most of their lives in trees, which are going to live on the trees. Okay, and they are going to conduct the essential activities on such as feeding, nesting, and even the reproduction on the tree itself. Okay, they are going to do most of the activities above the ground. Okay, those type of or that type of adaptation is called as arboreal adaptation. And this arboreal adaptation ke example gibbons. So these are the gibbons. These are going to live on the trees. Most of the life, most of the activities, and most of the life is going to spend on the trees. Now we will check the option. That option is present in the option D. So option D is the correct answer for that. Now moving on to the next question. The cofactor involved in the conversion of succinic acid to fumaric acid is option A NAD plus, option B FAD, option C GTP, option D NADP. They are asking question from Krebs cycle here. Conversion of succinic acid to fumarate. They are asking the cofactor. So how succinate is going to convert it into fumaric, by which cofactor? For this, you can see in the diagram here. This is the Krebs cycle diagram. And you can find out the succinate here. Succinate is going to convert in the sixth step. Succinate is converted into fumarate by the cofactor FAD, which is converted into FADH2. Which is the correct option here? Then FAD is present in option B. So option B is the correct answer. Moving on to the next question. The plants during salt stress tend to accumulate which of the following substances? Option A alanine, option B proline, option C oxalic acid, option D salicylic acid. The correct answer for this is option B. They are going to accumulate proline. Okay, they are going to accumulate the proline. Option B is the correct answer. Moving on to the next question, 72nd. BioNet International was initiated by option A CAB International, option B ICSU, option C WWF, option D UNEP. The correct answer for this is option A, that is CAB International. What is this BioNet? BioNet International, it is a company where it helps the other countries for the sustainable development. And that was initiated by the CAB International. Moving on to the next question. The most preferred organism for industrial production of recombinant insulin is option A Saccharomyces, option B Lactobacillus, option C Pichia, option D Bacillus. The correct answer for this is option B, that is Pichia is the correct answer. Moving on to the next one, 74th one. Antibodies obtained from hybridoma for one gene are known as option A polyclonal antibodies, option B monoclonal antibodies, option C multiclonal antibody, option D differential antibody. The correct answer for this, see, they are asking antibodies obtained from hybridoma for one antigen, that is known as what? Option A nalide poly anta ide. Poly means many antibodies. So they are asking here for one antigen. So haga option A wrong agutte. Mono means single. Okay, one antigen. That's why option B is the correct answer. Multi means many and differential antibody, no. These two are again wrong answers. Option B is the correct answer. Moving on to the next question, 75th one, last one. The scientist associated with work of genetically engineered bacteria for degradation by hydrocarbon is option A Joseph Lister, option B Anand Chakraborty, option C H.G. Horan, option D Inder Verma. The correct answer for this is option B Anand Chakraborty. Okay, that's about this video. So by this video I have completed the solving the paper 3 question paper from 2014. In the next video I will start with some other question paper. I'm hoping this video is going to help you all, and if you like this video, please do share and subscribe, and thanks for watching.